Hi, welcome to Bio24's lecture on pandemics. Pandemics are when there is an outbreak of a disease above normal or expected levels throughout the world. Now, when we say throughout the world, we really mean at least 3 or so continents. We don't literally mean it has to cover the entire globe. When we say that it's an outbreak of a disease, we say that the disease has to affect human health. And this is generally in reference to an infectious disease. At least that's what we'll be talking about with this lecture. An infectious disease is spread by a pathogen, or a microbe that causes diseases. The pathogen can be a bacterial cell, a viral particle, a fungus, or something else. Pandemics are usually spread from person to person, but can be carried on food items, or even inanimate objects. Pandemics are on the rise because of increased global movement. Historically, pandemics would be restricted to geographic locations. When cities were infected with the plague generally stayed within that city or that country because it was difficult to travel while sick, and it was also easy to quarantine different areas. Sick people move around a lot less in the past than they do now, mostly because travel 100 or 200 years ago was quite difficult, whereas travel today is very easy, and one can do it when a person is very ill. Today, global travel isn't just easy, but it's also very fast. So you could travel from place to place and not even know that you're infected with a disease until you get there. Not only is it easier for people to travel, but food and materials can also easily move from place to place, making movement of infectious diseases that much easier as well. Food items could of course be fruits or vegetables, but also farm animals. And this is another way that diseases are easily spread. What makes a good pandemic? An effective pandemic is easily transmitted from person to person, does not kill the infected person immediately. For instance, this is why Ebola, even though it's very deadly, generally tends to be restricted within a certain geographic region and is not so widespread as something like HIV, which has a very long incubation period, and a healthy individual can be infected with HIV for a much longer period of time before showing symptoms. Finally, another factor of a good or effective pandemic is if it's an infectious agent that can be transmitted on food or materials. This is one of the reasons why the flu virus is thought to be an effective pathogen that's easily prone to form pandemics. The flu, in fact, is something that we've seen historically be effective pandemics and is still something that we're concerned about today. The first big pandemic that we saw worldwide was in 1889. In 1889, a pandemic of the flu virus was spread around the globe. It was first reported in Russia and then spread to the Americas, India, and Australia. The reason why this was considered the first real pandemic is because prior to this, infectious disease agents would generally just be restricted to 1 or 2 continents. But the 1889 flu, as you could tell by the way that it spread, ended up moving to 4 or 5 different continents, covering a vast majority of the globe. It took 1 year for it to move all around the world and lasted over 10 years, but eventually died out after that. That particular strain of 1889 flu no longer exists today. The 1889 flu, much like other infectious diseases, demonstrate what's considered a U or J-shaped mortality curve. And you can sort of see this in the dotted line here. Mortality curves like this are mapped so that the age is on the x-axis and the death rate, or your chances of dying, are on the y-axis. Many infectious diseases, including many flu strains, kill the very young and the very old. But individuals who are in their 10s and 20s and 30s tend to not have to worry about dying from the flu. They just become very ill. This is, of course, with the exception of pregnant women. This is what's referred to as a U-shaped mortality curve because it takes on this characteristic U. There's also the J-shaped mortality curve, which is flat for young individuals and then rises for older individuals. J-shaped mortality curves just kill primarily older people and not the very young. This was different than our second pandemic that we're going to talk about today, and that's the 1918 flu pandemic. That is what's mapped here in the solid line. As you can see, the 1918 flu pandemic did not exhibit the U-shaped mortality curve so widely recognized in so many other infectious diseases. We're going to come back to this curve in a moment. This was the 1918 flu pandemic as photoed in a warehouse that was used in place of a hospital. It was first identified in Kansas Army training camps and was essentially a very short-lived pandemic, vanishing completely in only 18 months. It took just a few months to travel around the world and had burnt out in 18 months. This is a trend that we see in pandemics. They tend to travel faster as global travel and technology allows for the spread and movement of people to occur more efficiently. But because they travel faster around the globe and spread faster around the globe, they do burn out faster as well. Estimates claim that in 1918, over 25 million people died. This was equivalent to 2 to 5% of the world population at the time. I would like you to keep this number in mind when we go over HIV. So 25 million people dead in about 1 year from the 1918 flu. This was a very high mortality rate, and scientists wanted to know, why was it so deadly? They looked at a lot of the statistics at the time, and what they found was mapping different age groups that the 1918 pandemic exhibited a very different mortality curve than what we'd seen in the past. Here you have the highest mortality rate, so your highest chances of dying if you were infected with the 1918 flu occurred if you were between the ages of 20 to 29. If you were very young or very old, 50 or over, your chances of dying from the 1918 flu were actually lower than if you were in your 20s. Now, if you think back about what was happening in 1918, this was the end of World War I. Part of the reason why this pandemic spread so easily was because it was transmitted through military troops traveling around the world fighting in this world war. So one could say that maybe the reason why there were so many people dying in their 20s was because they were the ones that were carrying the disease. But what you have to understand about mortality rate is it's not the number of people that were dying from the disease. This was your chances of dying if you were infected with it. So your chances of dying from the 1918 flu were much higher if you were in your 20s than if you were very young or very old. In addition, because it was being carried by people in their 20s, they were also the ones that were dying at a much higher rate. This led us to form this graph that you see here, where your chances of dying, your death rate, was a lot higher when your immune system was in fact extremely healthy. Scientists wanted to understand why there was this discrepancy. This is quite scary because usually a lot of hospital efforts are dedicated to caring for the very young or the very old. And if a pandemic like the 1918 flu were to come back, that could end up really killing off a huge portion of the population, of the very healthy population in their 20s and 30s. So our military branch, specifically the CDC, which works under the military's guidance, the Centers for Disease Control and Prevention, to understand the 1918 flu pandemic and make sure that it doesn't ever happen again, wanted to investigate why this was the case. They ended up putting together the genetic sequence of the 1918 flu strain. Where they found this 1918 flu strain was from frozen cadavers found in cemeteries in Alaskan permafrost. Permafrost is permanently frozen ground. It's frozen year-round, and so there's very little to no decay of the body's tissues in permafrost. Alaska, being part of the U.S. territories, the CDC had control over and governance of decisions made in cemeteries in that area. So they went to cemeteries in Alaska in the permafrost and dug up some graves and extracted some lung tissue. And there they found the 1918 flu strain, kept frozen for almost 100 years. After sequencing the genes for the 1918 flu and analyzing the protein structures that it formed, they were able to better understand why it killed preferentially healthy young people in their 20s and 30s. They found that the way the 1918 flu worked is that it attacked the immune system by overstimulation of the immune system. This is what's referred to as a cytokine storm. A cytokine storm is a result of the flu virus releasing molecules that stimulates the immune system. These molecules are cytokines. And the cytokines, when they stimulate the immune system, cause an overstimulation of the immune system, resulting in the immune system not only attacking the flu virus, but also attacking the own body's tissues. When the immune system attacks the body's tissues, especially in the lungs, it causes fluid to build up in the lungs. When the liquid in the lungs is built up, it causes the patients infected with the 1918 flu to not be able to breathe anymore and to essentially suffocate and die. Now, this only happens if you already have a healthy immune system. People in their 20s and 30s with a healthy immune system are at a normal level of immune system function. So when the cytokine storm comes, it overstimulates the immune system, causing it to attack the body's tissues. But if you're in your teens or 40s or 50s, your immune system is weaker. And so when the cytokine storm hits, it essentially just increases the immune system so that it functions normally instead of being overstimulated, causing those individuals to not perish as a result of this overstimulation of their immune system. This is probably why the flu was able to attack mostly younger people and didn't affect the very young or the very old as much. In our next section, we're going to talk about a more modern pandemic, HIV.